Assalamualaikum and good day Dr. Zati. We are ARAYZ from Group 2, chose article Supercoiled DNA by William M. Scarver for our group presentation. Okay, so this is our group members. Now let's proceed to the content of our discussion. DNA is actually covered under the topic of nucleic acids, and in these slides, first we will discuss about topology and tertiary structure of DNA. Second, energetics and torsional tension in supercoiled DNA. Third, physiochemical characteristics, and fourth, implication for biological function.
So in topology and tertiary structure of DNA, we learn about the linking number, where it determines the topology and tertiary structure of DNA. In Watson-Crick double helix hypothesis, it stated that when alpha is equal to 36, means that DNA is relaxed, and this condition we'll assume as alpha naught. Tertiary structure of DNA formed when alpha is less than the alpha naught. It causes the number of base pairs protein increase and the angle between them decrease, and the underwound of DNA experiences the torsional stress, and this will lead to the formation of the tertiary structure associated with the supercoiled of DNA. This reaction is catalyzed by topoisomerase 1, where it increases alpha and relaxes the supercoiled DNA, and topoisomerase 2 or gyrase, where it decreases alpha and increase the negative supercoiling of DNA in ATP-dependent reaction.
Next is about energetics and torsional tension in supercoiled DNA. Supercoiled DNA is under torsional strain and have higher energy form than relaxed DNA. Below is the equation of free energy of supercoiling. There are some involvement of supercoiled DNA, such as in DNA replication. A major triggering factor for the initiation of replication is the supercoiling state of DNA. For instance, in E. coli, at a specific source of replication called ARAYZ, a supercoiled DNA molecule is required for the accurate initiation. The next one is in transcription. The movement of RNA polymerase will generate positively supercoiled DNA in front of polymerase and negatively supercoiled DNA behind polymerase. The positive supercoiling is then relaxed with the aid of topoisomerase to introduce net negative supercoiling DNA.
What is topoisomer? It is the DNA molecule with exactly same DNA composition but have different topology and linking number difference. As we carry out gel electrophoresis, the supercoiled DNA will have higher sedimentation rate compared to the relaxed DNA. Why? Because relaxed DNA has the highest medium resistance. Individual topoisomer can be isolated by extracting appropriate band, but topoisomer with the same number of supercoiled and opposite sign co-migrated. So, how to resolve this problem? By adding ethyl bromide in the agar gel electrophoresis, which tend to make negatively supercoiled DNA become more positively supercoiled. Consequently, mobility of previous co-migrated topoisomer of opposite sign become different in accord with their actual read.
Next, let's get into the general nature of supercoiled DNA. The torsional part of the supercoiled DNA may be reduced by some sort of complex transition. For example, the conversion of pDNA, the right-handed direction, to a zDNA, the left-handed direction, will remove approximately 2 supercoils and reduce the free energy of supercoiling. Furthermore, the behavior of supercoiled DNA is much more rather similar to single-stranded DNA. This is proven by the number of plasmids, such as E. coli plasmid, which is called L, shows a unique sequence for being a hypersensitive to S1 nuclease cleavage. This means that single-strand binding proteins have been shown to hint strongly to supercoiled DNA while exhibiting little or no affinity for relaxed DNA.
Today, I'll be presenting about the implication of supercoiled DNA for biological function. Torsional strain is caused by the unwinding of DNA double helix, and it is shown that torsional strain plays an important role in DNA mechanism. Firstly, the role of torsional strain can be seen in replication process in prokaryotic cells. For example, in E. coli, whereby the inhibition of DNA gyrase activity can affect the process of replication by preventing the relaxation of supercoiled DNA. It is shown that the efficiency of transcription by RNA polymerase increases when the negative superhelicity increases. However, some genes are maximally expressed on highly supercoiled DNA while some don't. In eukaryotes, torsional strain may act as a driving force for the altered conformation in eukaryotic chromatin and the resulting transcriptional selectivity for the gene sequences. Hence, we can conclude that DNA supercoiling can affect replication and transcription process. So that is all from me, and now we will proceed with the conclusion and questions.
That's all from our group. Thank you.